MUHAS and Kumamoto University was formalized in January 2016 when we signed the MOU. And the aim of this collaboration was to foster academic and research exchanges between the two institutions. And it was built upon the existing relationship that was already happening between MUHAS faculties and Kumamoto University faculties, and some of the very earlier students that happen to study at this center where Professor Ueno is coming from. His name is Dr. Philip Mwimanzi. He was 3 years ahead of me in medical school. And after that, several other Tanzanians, and especially MUHAS faculty, has benefited from that. So since then, the collaboration has led to successful research advancements in various areas. And the topics that have been covered include HIV drug resistance, HIV diversity and disease progression, HIV-related chronic inflammation, host restriction factors, and immune responses to SARS-CoV-2. And MUHAS has greatly benefited from this collaboration with improved molecular laboratory facilities through equipment donation and training opportunities that has been offered to our colleagues and our students. And moreover, the collaboration has successfully secured grants for HIV and COVID-19 studies that some of them have been completed, some of them are ongoing, and there are several other efforts to apply for new grants. Faculty exchanges has promoted knowledge sharing and expertise between the two institutions. Already, 6 Tanzanian scholars have completed their PhD studies at Kumamoto University, and partnership continues to offer internship and PhD opportunities to ambitious MUHAS graduates, as Professor Ueno mentioned earlier in the morning, those who are very, very competent. But we are very proud of the products that have come out of that. So overall, this collaboration has yielded significant outcome in research, laboratory strengthening, grants application, and knowledge exchange. Now, colleagues, ladies and gentlemen, in this session today, as introduced earlier on by Professor Mate, we will be having a number of very good sessions. So pay attention, ask questions, reserve some of the questions, and then after this session, try as much as possible to connect one another, and let's see what we can do in the future. Having said that, may I take this back to the chairperson of this session, Professor Mek Mate, to proceed with you. Thank you so much.
Thank you, DVC. So may I call upon now Dr. Doreen Kamori for your 20 minutes presentation, including questions and answers on a topic on HIV, the burden of HIV resistance in Tanzania, findings of the National Representative Survey. Karibu. You need this.
Okay, thank you very much, Chair, for the introduction. Good afternoon, everyone. So, as said earlier, I'll be taking you through the burden of HIV drug resistance in Tanzania during this era of dolutegravir use in the country. So just a brief background on the global statistics on HIV epidemics. We see that by 2021, it has been estimated that at least 38 million people are living with HIV, and 1.5 million people had acquired new infection, and at least 650,000 people have died of HIV-related death. However, the trend of the number of HIV-related deaths has been declining over the years. As you can see here, the data as early as in 2000, compared to the more recent years, there's a significant decline of the number of HIV-related deaths, and actually it's projected that by 2030, the number of HIV-related deaths will decrease to less than 400,000. And this is mainly attributed to the use of antiretroviral therapy globally. And Tanzania actually is among the country that has tremendously increased the scale-up of ART use among people living with HIV. As you can see here, in a span of 20 years, there's a 1,000 times increase of ART use in Tanzania. So although we are doing very well with regard to scaling up ART use, but there are several challenges that happen or faces the ART program in Tanzania. So these include emergence of HIV comorbidities, and more importantly, emerging HIV drug resistance, which subsequently has led to lack of viral suppression in some individuals, but also have affected the quality of life of people living with HIV, and has led to emerging drug resistance. This has also led to limit ART choices in our country. So there are several ART drugs that are in use in our treatment regimen. We have the NNRTIs, which previously were the default first-line regimen for treatment. And then we have the NRTI, which is the backbone ART drug for our treatment regimen in Tanzania. And of course, we have the protease inhibitors, which are used mainly in the second line and some population of children. But in 2019, Tanzania was among the country which rolled out integrase inhibitors, specifically dolutegravir and ratutrelvir. This was following a recommendation from WHO after seeing there's a high prevalence of resistance to NNRTI. So dolutegravir is actually a very good drug because there are several studies that have shown that this drug has high potency and is high tolerable and is highly effective in inducing viral suppression. And apart from that, it has fewer drug interactions, and most importantly, it has high genetic barrier to resistance in comparison to the other ART drugs. So apart from these drugs, of course, there are fusion inhibitors and capsid inhibitors, which are available in other developed countries. So to date, evidence shows that these all available ART drugs, they are actually susceptible or vulnerable to be resisted by HIV once the virus has acquired some specific mutations. And this resistance can either be pre-treatment, meaning that it occurs among people who have not been initiated to treatment, or can be acquired occurring among individuals who have been initiated to treatment. So what does the local data tells us on the burden of pre-treatment drug resistance in Tanzania? So we see that over the years, Tanzania had already crossed the threshold, the WHO threshold for pre-treatment drug resistance, having a prevalence of more than 10%. And in terms of acquired HIV drug resistance, the local data that is available also shows that there is a high prevalence of HIV drug resistance in Tanzania. So the next question was, what is the trend after dolutegravir rollout in Tanzania? We see that this is a good drug, it is highly effective, but how effective it is in our setting. So to address this, we conducted a national representative survey with two main objectives. One was to determine the estimate of prevalence of viral suppression among people living with HIV in Tanzania. And secondly, we wanted to estimate the prevalence of HIV drug resistance among individuals who are experiencing virological failure. So we had two main populations. We had the population of adults and children, who were further categorized into two main groups. Those who were on ART for a late time point, which is defined as being on ART for a period of 12 plus or minus 3 months. And then we have those who were on ART for a late time point, which is categorized as being on ART for a period of more than 48 months for adults and more than 36 months for children. So this survey was conducted actually 18 months after we transitioned to dolutegravir, and it involved 36 facilities that provide ART services in Tanzania. And these facilities were distributed in 22 regions in Tanzania mainland. So we collected some social demographic and clinical data from eligible participants, and we collected blood specimen, which was processed into dried blood spot, DBS, and plasma, and we estimated the HIV viral load. And for samples which were eligible for genotyping, were sent to a WHO accredited laboratory for HIV drug resistance genotyping. So in this survey, in total, we recruited 2,039 eligible participants, and adults contributed to 57.5%, and they had a mean age of 42 years with standard deviation of 12.4, and majority were female, while children contributed to 42.5%, and they had mean age of 9 years with standard deviation of 3, and of course, majority were also female. So after estimating the HIV viral load copies, we observed that only 137 participants were eligible for HIV drug resistance genotyping, having more than 1,000 copies per ml. And majority of this population constituted of children at the prevalence of 67.2%. So their samples were subjected for HIV drug resistance genotyping using the Sanger sequencing technique. And we targeted 2 major regions for HIV drug resistance. This is integrase region and the protease reverse transcriptase region. And the success rate for the sequencing was all above 98%. So with regard to the viral suppression, we saw that among the adults, 96 were viral suppressed, and among children, 89% of them were viral suppressed. But further stratification to a cutoff of less than 50 copies per ml of HIV viral load as recommended by WHO, which is the desired target, we see that by the time we were doing this survey, only 87.8% of adults had achieved this target of having HIV viral load of less than 50 copies per ml, and only 74.4% of children achieved this target. So further stratification of viral suppression versus ART regimen, we saw that 96.4% of adults on integrase inhibitors were viral suppressed, and 91.3% of children were viral suppressed, while those on second line, only 78.8% of adults and 85.4% of children were viral suppressed. So with regard to the prevalence now of HIV drug resistance among the eligible participants who had high viremia, we observed that 71.5% of these individuals are the ones that we could detect HIV drug resistance to either of the ART drugs, meaning integrase inhibitors, NRTI, and PIs. And most importantly, we saw that although integrase inhibitors, we believe that it's a good drug and it has high genetic barrier to resistance, but we detected about 5.8% of individuals had acquired integrase inhibitor drug resistance mutation. And also of note is that we also observed that about 44% of individuals had also drug resistance mutations to NRTIs, which is the backbone drug in our ART regimen. So this is just a further description of the mutations that we detected. With the integrase inhibitors, we have 2 major types of mutations. We have the major and accessory ones, which once they occur independently, they do not affect the drug effectiveness. At least that is what data tells us. So in this survey, we detected integrase inhibitor drug resistance mutation in 8 participants at 9 different positions. But of note is that we detected 6 different types of major integrase inhibitors drug resistance mutation, and these mutations have been shown that they are normally selected in vivo by dolutegravir. And here is the description of the other drug resistance mutation to other classes, such as the NRTIs, and PIs. We see that the most predominant mutation among NRTI is at position 103, and among NRTIs is at position 184. This is similar trend to both populations. So apart from the detecting this drug resistance mutation, we also performed some bioinformatic analysis to predict the genotypic susceptibility of these individuals who have high viremia and some have mutations. How are they susceptible to the available ART drugs? So here we have the data for both children and adults who are on first-line ART. So from this analysis, what we could be able to deduce is that we saw that majority of the individuals were actually predicted to be sensitive or susceptible to integrase inhibitors, with the exception of individuals who had these integrase inhibitor mutations. And what we also gathered from this analysis is that high level of resistance was predicted to NNRTI, about more than 60%, and commonly observed in efavirenz and nevirapine, likewise in the population of adults. Of course, this is expected, but we also saw that a high level of resistance is predicted to NRTIs, mostly the lamivudine and the mitristabine. And we also saw that high proportion of individuals are still susceptible to tenofovir, about 80%, and majority were also predicted to be susceptible to the PIs.
Hello. Mwambiaje jengo namba 4?
Okay, so among analysis among the individuals on the second line ART, what we could deduce from this prediction analysis is that we did not observe resistance to dolutegravir, both among children and adults. However, we also predicted again high level of resistance to NNRTIs and NRTIs. And we also observed that majority of the individuals are still sensitive to the PIs, and there was no cross-resistance to darunavir, which is the second generation protease inhibitor. So we also further stratified the analysis of patients who have integrase inhibitor drug resistance mutation to see their profile, especially those who have had all sequences available. So what we noticed is that by these all individuals were on dolutegravir-based regimen, and we also noticed that majority of them had multiple NRTI drug resistance mutation, meaning that these mutations might have already existed prior transitioning to dolutegravir, and therefore these individuals, when they were transitioned to dolutegravir, they were subjected to the DTG monotherapy. So what we can conclude from this survey is that HIV viral suppression among adults is above 95%. So adult population have actually achieved the third 95 goal of 1995 goal. And we also see that a large proportion of individuals with high viremia in Tanzania, they have drug resistance mutation. And of importance, we see that 5.8% of individuals have acquired integrase inhibitors drug resistance mutation. So this is very alarming, especially when you compare the data before transitioning to dolutegravir. One of our area study which we conducted using treatment naive individuals in 2017, we actually did not detect any major integrase drug resistance mutation. We detected only accessory at 1.8%. But we see that just 18 months after transitioning to dolutegravir, we already see that there is a major integrase drug resistance mutation at a prevalence of 5.8%. And actually, more data is emerging in other African countries in sub-Saharan Africa. For example, in Botswana, they have reported prevalence of dolutegravir resistance among individuals who are infected with subtype C at 30%. And then we have a couple of studies done in Malawi, which have reported the prevalence of dolutegravir resistance. Yes? Okay, sorry for that. And also in Uganda. So this is quite alarming to the ART program in sub-Saharan Africa. Thank you.
I allow 2 quick questions. Yes.
So do you think the transition to the integrase inhibitors was a little bit hurried? We could have a little bit of a waiting period before transiting to this? And the second question is, what is the future now? What is the hope on the table, like a last resort after a coming number of few years? I think the resistance to this type of medications will also be very high. So what does the future look like with respect to antiretroviral chemotherapy? Thank you.
Okay, thank you very much for your good questions. So actually the transition was not hurried. It was the right timing. But it's more on the strategy on how we transitioned. Because most of the guidelines in sub-Saharan Africa, they allow transition to all people living with HIV, okay, without considering HIV viral load status. So it means that there are some individuals who are experiencing virological failure, and as we have seen in this case, that they already have accumulating drug resistance mutation to NRTI, which is the backbone drug. So subjecting a number of people to DTG monotherapy, and therefore increasing the risk for dolutegravir resistance. So I think it's more on the weakness on the strategy of transitioning. And with regard to recommendation, as where we are, although dolutegravir is performing very well, but we need now to do more routine surveillance to see how we are going. And perhaps for those people who have not yet transitioned, then it's also important to at least do viral load monitoring so as to detect earlier individuals who are experiencing virological failure, and perhaps do HIV genotyping so as to select appropriate ART regimen for them, which will not increase the risk for emerging integrase resistance. Thank you.
Okay, I now invite the second presenter. In this case, it is Kasho Matsumoto. She's going to talk about activity of CD4 against various HIV-1 clinical strains. A brief introduction, presentation.
Thank you for attending my presentation. My name is Kaho Matsumoto, postdoc in Japan. Ninai wa Kaho, Nimefrafi Kukufaham. Asante ko Rika. It's my first time to visit Tanzania, and I had a lot of good time here thanks to my many kind Tanzanian people. So I like tangawizi, and my favorite food is beef mishkaki, and I got a henna tattoo. So yeah, I don't want to go back to Japan, to be honest. But yes, tomorrow I'm leaving. So let's start my presentation. My theme is activity of CD4 mimetic compound YIR821 against various HIV-1 clinical strains. How can I? Okay. As Dr. Doreen also showed in her introduction, 38.4 million people are globally living with HIV, and 28.7 million people are accessing antiretroviral therapy. HIV infection is a chronic disease which can be controlled by ART. However, there is no cure, and people need to access the treatment forever. Also, long-term ART caused various various difficulties such as appearance of escape mutant of HIV and medication adherence and high medical care cost. Moreover, some untreated individuals experience chronic inflammation leading to several comorbidities and accelerating aging. To solve these problems, we need the development of new treatments. So, yeah, okay. CD4 mimetic compound, CD4MC, bind to the CD4 binding site of HIV envelope, induced conformational change and induced conformational change in the envelope trimer, and has functional properties such as entry inhibition, enhancement of binding, neutralization, and ADCC activity by antibodies. So recently, we reported new CD4MC, YIR821, which has low cytotoxicity and high anti-HIV1 activity. So we examined the YIR821 has more potent activity compared to prototype CD4MC NBD556. And also we found that YIR821 has activities of entry inhibition activity, enhancement of binding, and neutralization activity against HIV1 isolate used in laboratories. And indeed, most previous research on CD4MC has also focused on these common laboratory viruses. So from this view, the objective of our research is to evaluate the effect of YIR821 on clinical isolates from various subtypes. For this purpose, we isolated envelope gene from patients' peripheral blood samples by PCR cloning method. At the same time, we extracted plasma IgG. Extracted plasma IgG from the same patient peripheral blood. Okay. So first, we analyzed about subtype B samples, which is common in the American, Europe, and Japan. We obtained 31 samples from Japanese patients infected with subtype B virus. And first, we focused on the entry inhibition activity of YIR821. We made pseudoviruses with envelope from each chronicle strain, and we analyzed the inhibition activity of YIR821 by TZM-BL cell assay. As I showed result here, 13 samples out of 31 subtype B samples were inhibited in the infection at the low concentration of YIR821, less than 50 micromolar. And we next examined the enhancement of neutralization activity by YIR821. We used TZM-BL cell assay here too. We compared the IC50 value of IgG in the absence and presence of YIR821. And enhancement of neutralization activity by YIR821 was observed in 15 samples of plasma IgG and 16 samples of 49C and 11 samples of 70B. And the result of IgG binding assay by flow cytometry is shown here. We used HEC293T cells as the envelope expressing target cell. We analyzed the enhancement of IgG binding activity in absence or presence of YIR821. The result is shown here. The slight enhancement of binding activity was observed in the case of plasma IgG because plasma IgG is polyclonal antibodies. However, in the case of monoclonal antibody 49C and 70B, which these antibodies against CD4-induced epitopes was significantly enhanced, were significantly enhanced in the presence of YIR821, significantly. Next, we assessed ADCC assay using clinical samples. To analyze the enhancement of ADCC activity by YIR821, we chose this assay system. HIV-1 infected CM-NKR-CCR5 cell is target cell, and NK cell line is as a effector cell. And to make the infected target cell, we made recombinant viruses. As we described on left side, envelope ectodomain from clinical isolate was inserted into NL43 backbone. We selected 6 samples that showed higher enhancement of POI9C binding by YIR821 from subtype B samples. So now we show the result of ADCC assay with autologous plasma IgG here. So we compare the ADCC activity in the absence and presence of YIR821 in the 6 viruses, viruses infected target cells. So as you can see here, all of the 6 samples, we can see the enhancement of ADCC activity by YIR821. Also, we analyzed the ADCC assay with monoclonal antibody. In this case, in this assay, we use anti-cluster A antibody A32 and anti-CD4 induced antibody 4IC, and the combination of these antibodies in the absence and presence of YIR821. Also, in this case, we observed the enhancement of ADCC activity by YIR821 in all 6 samples. So next, we examine the activity of YIR821 against Tanzanian samples, non-B subtype of Tanzanian clinical samples. So we obtained 55 blood samples consisted of subtype A, C, D, and these chimeric viruses as non-B subtypes. And the data of M3 inhibition assay using TZM-B cell is shown here. So y-axis indicates % coverage, and the red bar graph means inhibition of the infection by YIR821 at a concentration less than 50 micromolar. As you can see here, entry inhibition activity was observed in all subtypes. Although the number of samples is limited, some differences in the susceptibility to YIR821 were observed among subtypes. For example, the lowest is subtype C, 38%, and the highest is subtype D, 86%. Neutralization assay using TZM-B cell is shown here. Y-axis indicates the IC50 value of IgG-mediated neutralization. So, although the autologous viruses are resistant against plasma IgG obtained at the same time in the absence of YIR821, they became sensitive in the presence of YIR821. We observed this phenomenon all subtypes. So also the enhancement of binding activity by YIR821 was observed in all subtypes. And correlation were not observed among entry inhibition, enhancement of binding, and neutralization activity. So next, we analyzed crystal structure of YIR821 with GP120 to elucidate contact ratio of YIR821 against HIV-1 envelope. The left picture shows the YIR821 binds the CD4 binding site of envelope, and the light picture shows 3 kinds of CD4 mimics, CD4 MCs overlaid into one, and this graph shows the contact ratio of these CD4 MCs. YIR821 shares contact ratio with other CD4 MCs, but contacts wider range of the ratio. Clear correlation between the functional properties of YR821 with the M sequence of contact region was not observed by several analyses. So as summary, this study revealed that YR821 was effective to not only subtype B, but also non-B subtypes in the setting of clinical isolates. And the effect of YR821 was observed more than half of clinical samples, suggesting potential for clinical application of YR821. And lack of correlation between the functional properties of YR821 with the sequence variability of the contact residues by crystal structure analysis suggests that amino acid changes outside contact regions have an impact on the effect of YR821. And many researchers collaborate in helping this research. And asante sana sana.
I'll be interested to know if this YR821 is the first in its class or it's an improvement of the previous similar classes, and if there was improvement, what has been improved to attain these better low cytotoxicity levels and better activity profile. And this last question on the nature of interactions you presented on your last but one slide. Well, I'd like to know in this crystal structure you showed, what are the major form of interactions happening there between your ligand and the protein? Thank you.
Sorry, I don't understand. Second question, could you repeat?
I mean, can you please share what were the major interactions happening here between your ligand and this? You said it observed contact with a wider range of residues, so the amino acid residues on this protein you were sharing here. So what was the nature of these interactions?
Do you mean the main message of this? I mean, what I showed this one? Sorry. I mean,
between the YIR821 and your protein here, or your target here, the CD4 MSC.
Okay. So first CD4. Okay. So first, for first question, you mean you asked about the YIR821 present CD4MC and the prototype CD4MC. So yes, we derived from the prototype CD4MC NBD556, but this is made only for entry inhibitor, and this was not expected as an enhancement of neutralized antibody activities. Not only the binding CD4 binding site and then inhibit infection, but and NBD556 has a high cytotoxicity, so we cannot use this on the clinical. So that study about NBD556 stopped. But we generated many CD4MC from the NBD556 to try the less cytotoxicity and more potent activity, such as enhancement antibody activity. To help IgG antibody activity, like neutralization and ADCC or something like that. Because after CD4MC binds the virus, virus can make a conformational change to open, then antibody can access the envelope, virus more easier. So that's why it can enhance the antibody activity. And about the second question, about the structure, crystal structure, I showed that YR821 binds the CD4 binding site of the envelope. I showed how CD4 mimic binds, and we compare the prototype NBD556 and other CD4MC, and the graph shows that YR821 is green one. Green one is YR821, and it means contact residue is wider, it means it can bind virus more stable. I mean, more compared to other CD4MC. That's why we compare this one, and then we check the contact residue of GP120 residue and each amino acid here. Is it okay?
Thank you
Thank you.
very much. I now call upon Tosi Mwakandile from MUHAS. She's going to talk about hypertension and immune activation in ART naive patients and people living with HIV. A brief introduction of yourself, followed by a brief presentation.
Good afternoon. My name is Tosi Mwakandile. I'm a PhD candidate who is working on effect of low-dose aspirin on HIV infection. But today I'm going to present part of my PhD work on hypertension and immune activation in treatment-naive people living with HIV. So my work is supervised by Dr. Grace Shayo and Professor Eligius Liamuya from MUHAS. Okay, so this is going to be the outline of my presentation. So we all appreciate the tremendous job done by antiretroviral therapy. Before introduction of antiretroviral therapy, people with HIV suffered from a lot of opportunistic infections and infectious complications like tuberculosis and cryptococcal meningitis. But after introduction of antiretroviral therapy, these conditions have massively declined. And now people living with HIV suffer from the so-called serious non-AIDS events, like non-AIDS events like cardiovascular diseases such as hypertension. So cardiovascular diseases such as hypertension have become an important cause of morbidity and mortality among people living with HIV. In fact, it is reported that a quarter of the HIV-infected world population has hypertension. And in our sub-Saharan Africa, prevalences of up to 50% of hypertension in ART-exposed people living with HIV have been reported, and slightly lower prevalences in those who are ART unexposed. However, when you look at the figures, they're still high. The impact of hypertension in people living with HIV is higher than that in hypertensives in the general population, as it has been reported that hypertensive people living with HIV have more risk for cardiovascular events and all-cause mortality. And even among people living with HIV themselves, those who are hypertensive have more risk for cardiovascular events and all-cause mortality compared to those who are non-hypertensive. Okay, so different factors have been implicated in the causality of serious non-AIDS events in people living with HIV. As you can see, we have the virus itself as one of the causes behind the serious non-AIDS events. Again, we have cardiometabolic adverse events of the antiretroviral therapy and other factors, but I want you to notice the central role of the immune activation. Again, cardiovascular diseases, being one of the serious non-AIDS events, share the same factors in its pathophysiology. As you can see, we have the HIV as one of the factors behind cardiovascular disease. Again, we have the antiretroviral therapy, and again, our inflammation / immune activation is also one of the factors. So narrowing down to hypertension, which is one of the CVDs, we can see several factors involved. But again, you see the role of the virus in the causality of hypertension in people living with HIV. But again, we have the ART. And also chronic inflammation / immune activation is also one of the factors. So despite this current understanding of the pathophysiology of HIV-associated hypertension, the pathophysiology is not fully understood because most of the evidence comes from studies which were conducted in animals, and even the few studies which were conducted in hypertensive people living with HIV, the relationship between hypertension and immune activation markers was not extensively studied. For those reasons, we investigated the cellular and plasma markers of immune activation and exhaustion and their relationship to hypertension in ART-naive people living with HIV. So our study involved cross-sectional analysis of baseline data of 365 clinical trial participants, and the clinical trial was conducted at HIV care and treatment centers in selected hospitals in Dar es Salaam. So it was conducted at Mbagala Rangitatu Hospital, Mwananyamala, and Temeke Regional Referral Hospital. Our population, of course, was newly recruited ART naive, initiating ARV drugs, adults, people living with HIV, and we excluded those people who had contraindications to use of aspirin. So we excluded people who had previous intolerance or allergy to aspirin or any aspirin-containing products. We excluded those who were asthmatic, people who were predisposed to bleeding, people who were on chronic use of antithrombotic therapy, and people with active or history of peptic ulcer disease, and again, people with severe renal disease. We also included pregnant women, which in this case were a vulnerable population. So in our study, we measured both cellular and plasma markers of platelet and immune activation and exhaustion. All of these were measured using flow cytometry, and they were done in Kumamoto University. So for the cellular markers of activation and exhaustion, we used PBMCs, the peripheral blood mononuclear cells. And markers were gated from the CD4 positive and CD8 positive cells based on isotope controls using FACSDIVA software to define positive and negative populations. So this diagram here summarizes our gating strategy. So we first, from the PBMC, we identified a lymphocyte population. After getting the lymphocyte population, we went ahead to identify single cells among the lymphocytes. And out of the lymphocyte, we identified those which expressed CD3. And out of the CD3 positive cells, we got our CD4 positive population and the CD8 positive population. And we used these cells to see the expression of the markers of interest. So for example, this one is the CD4 positive population. Our marker for T cell activation was dual expression of HLA-DR and CD38. So this one is the CD4 positive population, which expressed both CD38 and HLA-DR. And again, for the marker of T cell exhaustion, we used PD-1 positive T cells. So this is a CD4 positive population, but these are the CD4 positive cells that expressed PD-1, which is our marker of T cell exhaustion. So the markers of CD4 and CD8 activation were expressed as frequencies of CD4 and CD8 expressing dually CD38 and HLA-DR, while for the markers of T cell exhaustion, we expressed as frequencies of CD4 positive and CD8 positive cells expressing PD-1. For the markers of monocyte activation and platelet activation, these were measured in plasma. So we used a customized BD cytometric bead array kit. The samples were acquired on the flow cytometer and analyzed by the FCAP array software, and the levels of the markers were expressed as picograms per milliliters. So hypertension was our outcome variable, and the predictor variables consisted of the markers of immune and platelet activation and exhaustion that we measured in our study, HIV-specific markers, that is CD4 count and viral load, and again, the traditional cardiovascular risk factors that are already established in literature. So what did we find in our study? So our participants had a median age of 37 years, and the majority were female participants, contributing to up to 65% of all the participants. So we had a majority of participants coming for ART initiation within 2 weeks of HIV diagnosis, and we had a significant number of our participants presenting with severe immunosuppression. These were ART naive participants, so majority presented with viral loads of 1,000 or more RNA copies per ml. So hypertension in our population was prevalent by 25%. We used the Mann-Whitney U test to compare the median values of immune and platelet activation markers between participants who were hypertensive and those who were not hypertensive. So as you can see from these dot plots, for all markers, the median values for hypertensives were higher than those for the non-hypertensives. So if you look at all these dot plots, except for soluble CD14, which was the marker of monocyte activation. However, the difference was not statistically significant. We also developed a generalized, so we also developed a generalized linear model to determine the predictors of hypertension. So all the predictor variables in our study so we also developed a generalized linear model for predictors of hypertension. At univariable analysis, we included all the predictor variables in our study, and those which had a p-value of less than 0.2 are the ones which were taken for multivariable analysis. Okay, so for the sake of this presentation, I'm only showing predictor variables which qualified for multivariable analysis. And as you can see from this table, none of the markers of platelet or immune activation predicted hypertension significantly. We just have chronic kidney disease staging and body mass index as significant predictors of hypertension. Okay, so what can we conclude from this? So although we have seen that none of the markers of platelet or immune activation predicted hypertension, we have seen that the median values of these markers were higher among hypertensives than non-hypertensives. Therefore, we recommend more studies to gather more evidence on this, and maybe the more studies should also study a wide range of the markers. So thank you very much and enjoy the rest of the conference. Okay. These participants whose data I'm presenting here are clinical trial participants involving the use of low-dose aspirin as the intervention. Therefore, we excluded people who had contraindications to use of aspirin. Any bias? Our trial was a randomized placebo-controlled trial, so we hope that we have taken care of that bias.
Okay, thank you. Responses in the lung of SARS. Brief presentation and then
good afternoon. I'm Catherine Mtali, first year PhD student at Kumamoto University. And today I'm going to talk to you about our project, Age-Dependent Pneumonia and T-cell responses in the lungs of SARS-CoV-2 infected mice. So to start with, COVID-19 caused by beta coronavirus, SARS-CoV-2, has become one of the worst pandemics of our time. So globally, there have been around 766 million cases and 6 million deaths as reported by WHO. And it appears that, as you can see, all ages of the populations are susceptible to SARS-CoV-2 infection, but this SARS-CoV-2 has exhibited age-dependent patterns in the terms of disease severity and clinical outcomes. So, however, the clinical manifestation differs with age, whereby older men more than 60 years of age at greater risk of severe disease, respiratory failure, and death. So these severe cases of COVID-19 are characterized by delayed onset of ARDS, as you can see around day 12, indicating that disease progression is driven by a dysregulated and destructive inflammatory immune responses rather than solely an inability to clear viral infection. So studies have been conducted and have shown that SARS-CoV-2 virus triggers an exaggerated immune response, causing release of pro-inflammatory cytokines such as TNF, interleukin-6, or interleukin-8. Also, there's a delayed production of type 1 and type 3 interferons, which they're supposed to confer antiviral protection and are critical for balancing immunity. But this late production, it enhances or promotes immunopathology. So this phenomenon is known as cytokine storm, but its fully mechanism is still not well understood. Also, together with other neutrophil and inflammatory macrophages that infiltrate the lungs, there's enhanced inflammation. So another study, which was done by our collaborators, which is like our base hypothesis, found that in the lungs of severe COVID-19 cases, there's abnormal T-cell differentiation or activation, but in normal Treg differentiation, as you can see, the APCs presents antigen. Do I have a question? Okay, APC presents antigen and it initiates T-cell activation, here via the interleukin-2 pathway. But so prolonged activation of T cells leads production of FOXP3 T cells, which suppress the T cell responses and hence immune T cell activation regulation or regulate inflammation levels. However, as I said, they saw that there's abnormal T cell differentiation initiated by TCR signaling, which can be associated with this COVID-19 severity. So this was like our base hypothesis. So recognition of exogenous antigens by T cell receptors is the upstream of the signals for antigen-specific T cell responses and can provide the initiation trigger for abnormal CD4 T cells phenotype associated with COVID-19 severity. So we ought to elucidate these temporal T cell signaling dynamics in between severe cases and mild cases. So based on the insight that I said, abnormal T cell activation and differentiation is associated with TCR, with mechanism of COVID-19 severity. So we aimed to study the involvement of the temporal dynamics of TCR signaling inputs in the mechanism of pathogenesis. So to study this, we established a SARS-CoV-2 infected mouse model with different disease outcomes with mild resistance disease and severe cases like severe pneumonia, and then use this to study the different temporal dynamics of TCR signaling inputs. So in order to study this TCR signaling input, we use NR4A3 Toki mice, which is a knock-in mice expressing a fluorescent timer protein gene as a reporter for NR4A3. So when T cell receives antigen here and upon recognition, TCR signaling induces NR4A3 fluorescent timer protein, which enables the in vivo analysis of TCR signaling. So the fluorescence timer changes color over time, over period of activation. So on early phases, it's blue, but post-activation, after completion of the inputs, it's red. So generally, the Wuhan strain is not, mice are not generally susceptible to the Wuhan strain. So as you can see, these are mouse AC cells, and there's no incorporation of viral protein into the nucleus compared to human ACs and other ACs. So in our study, we used a mouse-adapted SARS-CoV-2 strain, which is HQMUS, and not all mice are susceptible to it. For example, BAP-C mice, they died upon high doses of this mouse-adapted strain. But in our study, we used the B6 background mice, the C57BL6 mice. So we attempted to construct an experimental system of SARS-CoV-2 infected B6 mice, as explained. And it was age dependent. For the older mice, we used week 32, and younger mice, we used week 15 to week 16. And we speculated that older mice are developing pneumonia, severe and mild, resistant to the middle-aged mice. So below this presentation, you'll be seeing green, blue, red. It indicates different groups, as you can see. So we inoculated the mice with 30 microliter of 1 × 10⁵ doses of the mouse-adapted strain under anesthesia. So we followed this for 5 days while measuring body weight and looking for any signs. Then after day 5, the right lung was isolated for histopathological analysis, and with left lung we used cell staining and flow cytometric analysis. So our analysis was based on, as I explained, daily weight measurement, evaluation of pneumonia levels, cell isolations, where we had general cell population staining and specific T cell stainings. Also, we did histopathological analysis. So after infection, as I said, the red is older mice, 32 weeks, blue is middle age, as you can see here. So older mice had severe weight loss compared to middle-aged mice. Also, they had high pneumonia levels compared to the middle age. So we were able to establish a transgenic mouse experimental model to analyze the mechanisms of severity of COVID-19 pneumonia, which depends on age of mice. So these were mouse lung at day 5 sacrifice. This is the control group. We are not infected. And the infected group, as you see, older age, like almost 60 to 80% of lungs were inflamed compared to the middle age, where it's like 10 to 20% of lungs were inflamed. So these were, I said, this was a general population staining panels, and these were staining panels. So we did this to evaluate inflammation of mouse lungs through various infiltrating cells. I'll go fast. And upon various gating strategies, as you can see, we had alveolar macrophages, IMMs, T cells, and I'll take you fast here, sorry. Also, we had another analysis for fluorescent timer protein analysis in the lungs of the mild and severe, mild and old age mice, whereby this analysis provided evaluation of antigen specific T cell responses and activity of T cells with this color panel. So this was our, like we're checking the signaling inputs with these fluorochromes. So as you can see, this is with only the middle-aged mice, week 15 and 16. So upon timer blue and timer red, you can see this is the control group, and this is the infected group. There are high frequencies of timer blue and timer red. So as I told you, these are the timer blue, timer red is the marker for T cell receiving signaling inputs. So, so upon the general cell population staining, we observed that both old and middle-aged mice showed increased number of inflammatory cells in the lungs. So almost the same, except for the Tregs population, where it was high in middle age, the young group, compared to older age. But so both of the old that had severe and mild, the middle age had background of inflammation in the lungs. So there, due to this, there was no correlation between weight loss of the mice, as you can see here, and inflammatory monocyte-derived macrophages or PMNs. But there was negative correlation in the Treg frequencies and weight loss. So also T cell activation was high in older mice for some markers, but almost the levels tended to be still, like T cell activation was almost equally between middle and old age mice. So I'll take you briefly, like what are the roles of alveolar macrophages and pulmonary and what happens after infections. So macrophages are the major phagocytic population resident in most normal tissues, that is hemostasis. So we have two types, tissue resident, for example, alveolar macrophages, and circulating monocytes, such as IMM. So after infection, for example, and they have different roles. As you can see, they are first line of defense, immune defense in the lungs. So post-infection, we have loss of resident alveolar macrophages, and there's infiltration of IMMs. So this phenomena was observed also in our experiment. Also, we have T cells. We know we have different type of T cells. So cytotoxic T cells, which are specialized to kill cells infected with intracellular pathogens, most notably viruses born to MH class 1. Also, we have CD4, which differentiates to different types, T helper 17, 1, 2, and regulatory T cells. But we're more interested in regulatory T cells, which are, as I told you, control and counteract excessive immune responses. So from the first part, conclusion is that, as I told you, both middle-aged and old mice, they showed same level of inflammation, cell filtration in the lungs, and gradual T-cell activation, both groups. However, there was a difference in the level of inflammation. So as I told you, young had mild pneumonia, where old had severe pneumonia. So we want to know what are the factors that are associated with suppression or enhancement of inflammation in the lungs. So as I told you, we did a detailed T-cell analysis where we wanted to know frequency of timer blue reactive T-cells in the lungs. So there was a significant, as I told you, in the timer blue, there was a significant differences in TCR signaling input dynamics of T-cells in the middle-aged and old-aged mice. So as you can see, middle-aged had received TCR signaling input compared to old-age mice. So, and body weight loss and reactive T-cell frequencies were inversely correlated. So from this, the first part I already mentioned, so in the middle-aged mice, reactive T-cells, T-cell signaling input were highly induced, suggesting that T-cell responses might contribute to suppression of lung inflammation. Also, and as we saw, a different phenomenon in old mice where there was insufficient T-cell responses due maybe to aging of the immune system, which may be the cause to the disease severity. So in summary, our study suggested that induction of effective Treg cells populations in the middle-aged mice are strongly associated with regulation of SARS-CoV-2 pneumonia. Also, as the phenomena I explained, old age had severe pneumonia and severe lung inflammation. So we want to know what are the causes of these, and they also had insufficient T-cell responses. So we wanted to know what are the causes of this TCR, insufficient TCR antigen signaling input. Is it age-related decrease in TCR diversity or changes in TCR signaling pathways or Treg function itself, age-related changes, or is it factors associated with antigen presentation? So we are conducting a detailed analysis of the T-cell responses. I would like to acknowledge people who collaborated in this study. Also, great thanks to my sensei, Takushi Nomura, and my fellow lab mate, Risa Kuroka. Thank you very much.
Thank you for a nice presentation. I'm interested to know the expected lifespan of this particular strain of mice you used. And then I will ask the second question. So how long are the strains expected to live in total?
Strain?
These mice, yeah.
Okay.
Because that was, I'm asking what was the basis for establishing this, for example, you are saying middle age, and then you say 32 was the oldest group. And then in some cases you do not see differences between your middle age and the oldest mice because you ended up 32. So literature can show some mice can live up to, for example, 18 months or 26 months to 30 months. So is 32 weeks enough to categorize them as old mice, or you could have gone higher in terms of age and get something different? So just a short question and comment. Thank you.
Okay, so maybe I say this. Most of experiments they do in young, maybe week 8, week 9 mice. So we thought week 32 was old enough. So I can't tell you how much they live exactly, but sorry.
So I now welcome Lillian from here. Lilian Mkenda. She's going to talk about neutralizing antibodies. Oh. Introduction while you're waiting.
Good afternoon all. So my name is Lilian Mkenda. I'm a faculty at the Department of Microbiology and Immunology and a second year PhD student. And I'm here to sort of narrate a story of what we all experienced in terms of SARS-CoV-2 pandemic from a perspective of immune responses after either infection or vaccination in Dar es Salaam, Tanzania. So this was us in 2020. We were all in a state of panic and we had more questions than answers. And at this particular point in time, what was at the top of the agenda was ascertaining control. Control, one, over the spread, and two, preventing newer infections from taking place. And so as you see different regional representatives here discussing, and I believe this is where we instigated measures like lockdown and vaccine manufacturing that later on proved effective. And so it was paramount that each and every particular region contributes to the gap of knowledge that we get the best way forward. 2 months post the pandemic, we almost had information overload. We knew the epidemiology, we knew the etiology, we're now knowing who was most vulnerable, and we even had vaccine prototypes. And of course, the cases continued to rise to what we currently have, and that is 700 million cases and 6 million cumulative deaths. What was important, or rather what was different, was the fact that the pandemic sort of differentiated the African region compared to the other regions globally. We had lower number of cases and deaths compared to what was reported elsewhere. And different speculations came about, and as a researcher and an African, I expected Africa to sort of spearhead on answering the narrative and perspective of what was really happening locally. However, this was not the case. In terms of contribution, of the output at this particular point in time, which was 2 months post the pandemic, the African region had contributed only 3% to the global output of the information that was being provided concerning COVID-19. And unfortunately, also furthermore, the majority of the contribution, almost 70%, were from countries like South Africa as well as Nigeria. Tanzania contributed 2% to the 3% pool of information, which is very slim as compared to the strategies that we took on. Now, if we reflect back to the pandemic, to a larger extent, we imported knowledge, we imported vaccines, we even imported the strategy on how to curb the spread of infection, and this included the instigation of lockdowns. Unlike other African countries, Tanzania was different on this particular aspect. We did not conduct lockdowns, and this could have some significant input on the type of immune response that we later on generated. And also, because of high levels of vaccine hesitancy, we also had lower vaccine coverage compared to what other regions had. So when you look at all these diverse differences, you tend to wonder, or you get interested. So what was the outcome? Did we now attain the herd immunity that we're looking for? Did we attain the protective titers that we're trying to achieve by not locking down and etc.? Considering the fact that there are different literatures available online that have shown past variants with unique mutations that had very low global prevalence compared to what was reported elsewhere, and also a 10 times mutated SARS-CoV-2 that was reported in 2021 compared to what was circulating at that particular point in time. And in terms of serosurveillance, also the country, for the countries ranging at 58 to 88%, I believe currently we have a higher seroprevalence rate. However, this information is not yet published. What is paramount at this point in time was understanding are the antibodies that we're reporting here post what measures that we had taken in terms of not looking down and our vaccine coverage. Are we protected? Have we mounted protective titers, and etc.? And these antibodies able to cross-react against different variants that have circulated and are continuing to circulate. This is very important because as the virus continues to evolve, it's important for us to know if we are on the other side of protection or we are within the globe of protection. So moving on, 3 years post-pandemic, we've now entered into the era where COVID-19 has ceased from being a public health emergency, and the key recommendation is continued conduction of serosurveillances, which is key to informing on the current and the future strategies in case we have another pandemic. But I would like to highlight what is key at the same time, that COVID is no longer a public health emergency. We have an immune escape Omicron variant that has had circulated in the recent past, and this being the XBB 1.5 that has shown to sort of escape immune responses even after 3 to 4 shots of the vaccine. Now you can imagine for a population that less than 10% have at least received 1 dose of the vaccine. So it's important for us to understand at what state are we in. And so this was the objectives of our study, to evaluate the neutralizing activity against the Omicron sublineage after infection and or vaccination in Dar es Salaam, Tanzania. The Omicron sublineage is the latest and the current circulating variant that is at hand. So we conducted a cross-sectional study at Muhimbili and Aga Khan. We excluded patients who were HIV infected and as well as patients who had malignancies. And therefore from here we acquired 3 groups, and these were those who were infected, those who were vaccinated, and those who were infected vaccinated. Moving on to the lab, in a nutshell, this is what we did. We had 2 sets of cell lines. The first cell line was used for viral assembly here, to where we assembled, we used the HIV backbone as well as the spike protein of the different variants. We also had the other cell line that was used to transfect cells with the important receptors that are essential for viral entry, and these are the ACE2 receptors as well as TMPRSS receptor. 48 hours post, we assembled the virus, the cells expressing the ACE2 receptors as well as the 5 times serial dilution plasmas. And we're expecting to see to what extent this plasma is going to inhibit entry of the virus into the cells that were expressing the ACE2 receptors. And see, this is what we did in the lab. So coming to results. So we found being older shown a 2 times increase in terms of cross-neutralizing antibodies. Also, we observed individuals who were obese, okay, having almost 2 times increased cross-neutralizing antibodies compared to their counterparts, and this could be explained by the fact that maybe most of these succumb to severe infection, because severe infection is likely to lead to higher levels of response. Chronic disease as well as severity of infection tended to become significant but did not achieve statistical significance. There was no difference in terms of duration, whether infection or vaccination, for those who were infected above 6 months and those who were infected below 6 months as per WHO recommendation. The WHO says that vaccine immune response tends to have reduced, by the 6 months mark, while the infection immune response by the 8 months mark. So we used the 6 months cutoff to sort of see if there's a difference between the 2 groups. Okay, so moving on the declining neutralization titers. So like I said, we had 3 groups, and in this particular chart we are seeing the NT50. This is the neutralization titers that we measured, and we measured comparing it against the variants. And here we have variants that we had experienced, the wild type and the BA1. These were variants that my particular population had experienced, and BQ and XBB, these were variants that were not experienced by this patient, and therefore we're we're now measuring to see if the antibodies generated are able to cross-react against the different variants. And as you can see here, what message I want you to take here is the fact that those who were vaccinated had a steeper decline in terms of cross-reacting antibodies. As you can see, the 3.2 times, the 15 times, and the 37 times compared to the other two groups. And also, we also had individuals who had absent neutralizing antibodies, and as you can see, this was more pronounced on the unexperienced variant. As you can see, those who were vaccinated, more than 50% were not able to attain the anti-50 for the XBB variant. So we went ahead and compared the three different groups, and that is the vaccinated, the infected vaccinated, and those who were infected only. And as you can see here, the green group are those who were infected vaccinated. Across the variants, they had higher median neutralization titers compared to those who were vaccinated and those who were infected alone. And the effect was uniform throughout. And what was interesting was the fact that these particular individuals had higher median titers, even for the higher median neutralization titer, higher than the cutoff, even for the variant that they had not experienced initially. And this was higher also compared to those who were vaccinated and infected only. This is important because when we consider a study that was published by Wang et al., we see that we sort of compare to this particular population here that had received 3 shots of the original vaccine and also had an added bivalent vaccine. As you can see, the median neutralization titer is slightly higher than the cutoff as compared to those who had received 3 shots and 4 shots. Now, if we had to put in perspective, mind you, a participant had only received 1 shot of the vaccine, and that is a Janssen shot. So we went further and scrutinized the infected vaccinated group. And here the infected vaccinated group, we grouped them into 2. We had individuals who were vaccinated and then got infection, and this is what we call the breakthrough infection. And then we had the second group that were infected and later on went to vaccinate. And here we see those who got vaccinated and later developed infection had higher median neutralization titers. Thank you. I apologize for that. So we went further to scrutinize the neutralization titers with respect of the particular dominant variant that was circulating at the time of data collection. And as you can see, we compared those who were the same group of infected vaccinated, those who were infected, those that we collected during the B4-5 wave and those that we collected in the B2 wave. And as you can see across the groups, with the exception of the BA1, the BA5 had higher median neutralization titers compared to those who were infected with the BA2 wave. So we went further and sort of further categorized the individuals who had breakthrough infection now. These were all infected vaccinated, and these were individuals who had breakthrough infection only. And we did the same scrutiny where we sort of grouped them according to the type of variant they had experienced at the time of data collection. And these are the ones that had experienced B4-5 wave, and the blue ones are the ones that experienced B2 wave. And although there were fewer, you still see a trend of those who had experienced the B4-5 wave had higher median titers compared to those who had experienced the BA2 wave. And this was statistically significant even for the unexperienced variant. So in conclusion, we see some sort of a benefit of the vaccine, and where we observed those who were infected, vaccinated had a better comparative advantage compared to those who were infected and vaccinated only, and those who suffered breakthrough infection were observed to have better responses compared to those who were infected and then got compared to those who were infected, yes, and then got vaccinated later. Thank you very much. I would like to acknowledge my supervisors, Professor Balandia, Professor Lamuya, Professor Ueno, for allowing me to conduct this lab analysis in your lab, and as well as the other funders. Thank you.